Good afternoon. I will conduct today's lesson. My name is Mусагулова Bahıt. I am from Nazarbayev Intellectual Schools in Kokshetau. And the lesson's topic of our lesson is biotechnology, cloning of plants and animals. The lesson's objectives of our lesson are describe the methods of plant micropropagation, explain the mechanism of cloning, compare features of plant and animal cloning, discuss the issue of cloning from an ethical point of view. And in the first, I give you some keywords that we use in our lesson. Your task is to say letters in right order in these words. At this slide you see the words, and each right answer of this task. And what is a clone? A clone is any organism genetically identical to another organism. Now, the creation of animals and plants with a specified quality has always been very tempting because it is meant to create a unique and need organisms that are resistant to disease, climatic conditions, giving sufficient litter, the required amount of meat, milk, fruits, vegetables, and other products. The use of cloning technology involves a unique opportunity to receive phenotypically and genetically identical organisms, which can be used for a variety of theoretical and practical problems facing biomedicine and agriculture. Now, cloning used for cloning plants and cloning animals. How we grow the plants we want. For this, we know how many types of plant reproductions. It's two types of plant reproduction. They are sexual reproduction and asexual vegetative reproduction. And what is the disadvantages of sexual reproduction? The disadvantages of sexual reproduction are, first, plants raised through seeds have long juvenile period. It takes long time to fruiting. Second, the seedling trees have very deep root system. Hence, the trees attain big size at maturity. Some viruses are seed-borne, for example, mosaic in peaches and psoriasis in citrus. These viruses perpetuate through seed. Some seedless fruits cannot be propagated through seed. The only method to produce these plants will be a sexual method. The improved characteristic of a particular cultivar cannot be perpetuated in plants raised from seeds. And the second way is vegetative reproduction. Vegetative reproduction, the genotype of the mother plant remains, as well as reducing the duration of the juvenile period. When a gardener has taken cuttings of this plant, which probably has good characteristics, and is growing them in a damp atmosphere until the roots develop. This is a cheap way of producing quite a lot of plants. What is the disadvantage of vegetative plant reproduction? The disadvantage of vegetative plant reproduction: offspring produces close to the parent to produce dense population, which may lead to competition for nutrients like ACT, no genetic variety, inbreeding, reduced biodiversity. If environment changes, all organisms are at risk of extinction. But now most of people use tissue culture. A tissue culture method when a few cells are taken and placed on nutrient agar until roots and shoots develop. They can then be planted normally This is a very efficient way of mass producing your perfect plant, but it is more expensive and difficult than taking cuttings. And now I give you three situations, and for each situation decide whether taking cuttings or tissue culture would be most appropriate and why. You decide this task in groups. And the first situation with Mother Hubbard. Mother Hubbard is on holiday with her family when she sees a lovely dahlia plant. She wants to grow a few of these lovely plants for her front window box. And what should she do? The next situation with Mrs. Foxglove. Mrs. Foxglove has a florist on the high street. In 6 months' time, she has a wedding that has ordered 12 magnolia plants. She has 2 plants in at the moment, but the plants are very expensive. And what should she do? And the first situation with Farmer Smith. Farmer Smith has a wonderful banana plant. It grows ripe, juice fruit that is large and does not bruise easily. A company in Spain would like to buy 500 of these plants at very good price, but Farmer Smith only has 200 of his own. He does not want to give them up. And what should he do? I give you a few minutes to discuss in groups these 3 situations and what of situation you use. And what is plant tissue culture? Plant tissue culture is a collection techniques to grow plant cells, organs, tissues under sterile conditions. Plant tissue culture is widely used for the in vitro vegetative propagation of plants in a process known as micropropagation. If we give definitions of these words, it's the in vitro and aseptic cultivation of any plant part of nutrient medium. For example, cell culture, tissue culture, organ culture. An essential aspect of plant biotechnology, genetic inbreeding of plants occurs at the level of single cell. The cell must develop into a whole plant through cell and tissue culture for applicable uses. What is explants? Explants an exact piece of tissue or organ taken from the plant to initiate a culture. Sterilization, procedure for the elimination of micropropagation. And the callus, it's an unspecialized and unorganized growing and dividing mass of cells. During callus formation, there is some degree of dedifferentiation both in morphology and metabolism, resulting in the lose the ability to photosynthesis. It has two habitations, compact callus in the first picture and friable callus in the second picture. Basis for plant tissue culture includes there are 2 hormones affect plant differentiation: auxin that stimulates root development and cytokinin that stimulates shoot development. Generally, the ratio of these 2 hormones can determine plant development. And you see, when auxin higher, cytokinin low, it's root development. When cytokinin high or auxin low, it's shoot development. And when auxin and cytokinin same, it's a callus development. Growth regulators are hormones that use the auxin, a group of plant hormones, natural or synthetic, which induce cell elongation or, in some cases, cell division. Cytokinins, a group of plant hormones which induce cell division and often adventitious shoots, and in most cases inhibit adventitious root formation. This picture illustrated control in vitro culture with cytokinin and auxin. And the factors affecting plant tissue culture. First, growth medium, minerals, growth factors, carbon source, hormones. Next, environmental factors, light, temperature, photoperiod, sterility, medium. Explant source. Usually the younger, less differentiated the explant, the better for tissue culture. And genetics and different species show differences in amenability to tissue culture. In many cases, different genotypes within a species will have variable responses to tissue culture. Response to somatic embryogenesis has been transferred between male cultivars through sexual hybridization. And three fundamental abilities of plants: totipotency, the potential or inherent capacity of a plant cell to develop into an entire plant if suitably stimulated. It implies that all the information necessary for growth and reproduction of the organism is contained in the cell. The differentiation capacity of mature cells to return to meristematic condition and development of a new growing point, followed by redifferentiation, which is the ability to reorganize into new organ. Competency, the endogenous potential of a given cells or tissue to develop in a particular way. And why we use this method? Because the production of clones of plants that produce particularly good flowers, fruits, or have other desirable traits. To quickly produce mature plants, the production of multiples of plants in the absence of seeds or necessary pollinators to produce seeds. The regeneration of whole plants from plant cells that have been genetically modified. The production of plants in sterile containers reduces disease transmission, allows production of plants from seeds that otherwise have very low chances of germinating and growing, i.e. orchids and nepenthes. To clean particular plants of viral and other infections, and to quickly multiply these plants as clean stock for horticulture and agriculture. And how we grow plants with this method? Adult plant cells are totipotent, meaning they have the ability to give rise to a fully differentiated plant. Because of this, it is possible to collect cells from a mature plant and use those cells to produce clones of that plant. Cloning in plants. Tissue cell culture, the first tissue sample scraped from parent plant, then these tissue samples placed in agar growth, medium containing nutrients and auxins, then samples developed into thinner plantlets and plantlets planted into compost. Types of in vitro culture: culture of intact plants, seed and seedling culture, embryo culture. Organ culture includes shoot tip culture, root culture, leaf culture, anticulture. And next, callus culture, cell suspension culture, and protoplast culture. On this picture you see types of stem cuttings which are used in vitro and types of leaf cuttings which are used in vitro. Plant tissue culture divided in two ways. First, embryogenesis, direct embryogenesis, indirect embryogenesis, organogenesis, organogenesis via callus formation, direct adventitious organ formation. somatic embryogenesis, a process in which embryo-like structures are formed from somatic tissues and developed into a whole plant. For somatic embryogenesis, embryos form which can develop and then germinate into a whole plant. This picture illustrated indirect somatic embryogenesis in carrot. The production of embryos from somatic or non-germ cells usually involves a callus intermediate stage which can result in variation among seedlings. Somatic embryos are complete entities similar to seeds. They possess a shoot and a root tip and have a complete vascular system. They form under specific conditions that are different from shoot morphogenesis. An auxin is usually applied for induction of somatic embryogenesis. Nitrogen and carbohydrate balance are also important. Embryos usually develop once they are transferred to a medium with no auxin. And this picture illustrated soybean embryogenesis. The first starting material, immature seeds, then induction, proliferation, development, and germination, and plant recovery. The advantages of this method: an almost unlimited number of plants can be obtained from one donor plant within a short period of time. Amenability to automation potential for mass producing genetically improved somatic embryos at reduced costs. Problems: growth response is highly genotype dependent. Declined regeneration ability after prolonged subcultures. Uncertainty about genetic variation using seed-derived explants. And the next way, organogenesis, when the production of roots, shoots, or leaves, these organs may arise out of pre-extinct meristems or out of differentiated cells. This, like embryogenesis, may involve a callus, intermediate, but often aqueous without callus. One of the methods of this way, shoot morphogenesis. Shoots can be formed directly from the starting plant material or arise from a mass of proliferating unorganized piece of plant tissue called a callus. Shoot formation is usually induced following placement of tissues on media containing a high cytokinin to auxin ratio. Media that contain inappropriately higher levels of auxin can cause callus to form. Plant regeneration from shoots regurgitates the formation of roots on excited shoots. Root formation can be spontaneous or induced by rooting hormones which contain auxin. Rooted shoots can then be transferred to soil for plant establishment. Somatic embryogenesis and organogenesis, both of these technologies can be used as methods of micropropagation. Not always desirable, because they may not always result in population of identical plants. The most beneficial use of somatic embryogenesis and organogenesis is in the production of whole plants from a single cell or a few cells. And micropropagation, the art and science of plant multiplication in vitro, usually derived from meristems or vegetative buds without a callus state, tends to reduce or eliminate somaclone variation, resulting in true clones, can be derived from other explant or callus, but these are often problematic. And the micropropagation includes few stages. The first, preparative stage, when donor plant selection and explant preparation. Explant quality is influenced by the physiological conditions of the donor plant. And starting material of micropropagation, they are tip bud, leaf, axillary bud, intermode, root. Next stage, establishment of explant in culture. Surface sterilization of explant tissues, isolation of explant under sterile conditions. Medium must contain all components necessary to make the explant perform as desired. Next stage, multiplication and proliferation of axillary shoots, repeated enhanced shoot production by high cytokinin in the medium, alone or with a smaller amount of auxin. Shoots must be transferred to fresh medium at regular intervals. Next stage, pre-transplant rooting. It is rooting of shoots or shoot clusters in vitro. Auxins are important for root initiation in vitro. Advantages of rooting after removal from culture includes reduced costs, structurally and physiologically better, avoid damage to roots and that occur during transplanting. And the next stage, transfer to natural environment. The last stage of the process of micropropagation, test to plant adaptation to non-sterile condition, is the most responsible, and its underestimation may lose all carried out hard work. Development a whole system test tube adaptations of plants to the usual conditions. First test tube rooted plants are transplanted into a liquid diluted with culture medium. Then about a week later, these plants are transplanted into autoclaved ground and to maintain a high humidity covered glass vessels. After 70 days, the cover is removed is maintained for about a week, even as special boxes, and only then are planted in greenhouse. This is a meristem tip culture that used in micropropagation. And the hitches of micropropagation, a clonal reproduction, way of maintaining heterozygote. Multiplication state can be recycled many times to produce an unlimited number of clones. Routinely used commercially for many ornamental species, some vegetatively propagated crops. Easy to manipulate production cycles, not limited by wild seasons, environmental influences. Disease-free plants can be produced, has been used to eliminate viruses from donor plants. And the advantages of micropropagation. From one to many plants rapidly. Multiplication is in controlled lab conditions. Continuous propagation year-round. And disadvantages of micropropagation, equipment facility intensive operation, technical expertise in management positions, protocols not optimized for all species, it may too expensive. And the next method, embryo culture. Embryo culture, development from the need to rescue embryos from white crosses where fertilization occurred, but embryo development did not occur. These techniques have been further development for the production of plants from embryos development by non-sexual method. this picture illustrated embryo culture. And now you see this picture, 6 plants: cactus, cotton, sunflower, poplar, soybean, and oxybuck Y. And which are somatic embryos, which are shoots embryos. 3 of plants shoot morphogenesis, and 3 of these plants are somatic embryo. And now we have a lab activity with cauliflower cloning. You have 2 tasks: produce your own cauliflower clone using the following method. Produce a step-by-step cute flow chart on how to use cuttings, tissue cultures to produce a clone. Extra challenge: when would plant cloning be useful?
Laboratory work is called cauliflower cloning. We are going to clone cauliflower using aseptic technique. First of all, we should sterilize workplace using ethanol. Like this. Clean everything. After sterilizing the workplace, we should sterilize equipment and other stuff using Bunsen burner like this, in order to get rid of bacteria and viruses. After that, we should take a small piece of cauliflower like this and cut it. And take the next part from it. And also sterilize it in sterilizing solution. We'll just shake it in a second and sterilize. After that, we should put it into growth medium, which is called agar. This growth medium helps to clone cauliflower and put it here like this. After that, there will be, after a few weeks, there will be new cauliflower.
And the next cloning use in animals, with animals. Cloning in animals is more difficult than in plants, but it can be done in 2 ways: embryo transplant and adult cell cloning. And the first, embryo transplant, you see in this picture. With methods of cloning: embryo splitting, artificially splitting a single embryo at a very early stage of development. In the natural process, this would create twins. However, because this is done at an early stage and 3 are usually less than 8 cells, you can only make a few clones. Both the nuclear genes and the mitochondrial genes would be identical. Adult cell cloning, applications of animal cloning: clone ship Dolly. In March 1997, a scientific breakthrough occurred. Jan Wilmot of the Roslin Institute in Edinburgh, Scotland, announced that he and his colleagues had cloned a ship from a cell taken from an adult cell.
Dolly is without question a normal, healthy, genetic marvel. She's an identical copy of an older sheep.
People expected that maybe at one time, at some time it would happen, but maybe we've been a little bit quicker than they expected.
Dr. Ian Wilmot and his team of Scottish researchers began with a 6-year-old sheep. They took a single cell from her mammary gland, including its nucleus, the command center containing all the genes, the DNA, that determine the sheep's individual physical characteristics, eyes, nose, hair. Then they took an egg from a second sheep, removed its nucleus, and replaced it with a cell from that first sheep. The egg and its new nucleus then fused together, turning into an embryo, but with all the genetic characteristics of the first sheep and none of the second. In effect, a carbon copy, an exact duplicate of the first. All that was left was to put this developing embryo into a third sheep, a host sheep, which carried it to term.
What is the mechanism of sheep cloning? This lamb has no father, but has 3 mothers. Sheep breed Finnish Dorset, which gave its genetic material. Scottish sheep breed Chernomort, from which took the egg, ovum of which haploid nucleus removed and placed in the cytoplasm of the cell without a nucleus diploid nucleus from a cell of the first sheep. And the third sheep, also the recipient, breed Scottish Chernomort that endured for 148 days of the farmer's lamb. This picture illustrated adult cell cloning. Dolly the sheep. You see 3 sheeps. First, a donor cell is taken from a sheep's other donor nucleus. Second, an egg cell is taken from an adult female sheep, egg cells. Then these 2 cells are fused using an electric shock. The fused cell begins dividing normally. Embryo grows, and then embryo is placed in the uterus of foster mother, and embryo develops normally into a lamb, Dolly. And who is her real biological mother? Ones whose genetic material has been introduced. How do you think about these technically complex experiments? It's difficult. The 256 attempts to transplant eggs succeeded only once. How does cloning off? From the egg, nucleus is removed. Then, from a somatic cell of the body, which will be cloned, allocate nucleus. Next, in the resulting egg, injected nucleus containing DNA cloned body. Egg begins to divide, and the nucleus is activated. A week later formed the embryo of about 100 cells. Then we use 2 ways of cloning. First, surrogate cloning, putting the embryo into the uterus of another organism, or second, therapeutic cloning, placing the embryos in the laboratory culture medium. And now I give you tasks. You write in the table what are the advantages and disadvantages of animal cloning. It works in pair. The advantages of animal cloning. First, we can make copies with the characteristics we want. We can make many copies of the kind of animal we want quickly. Extinct or endangered species can be repopulated through cloning. Makes it possible to produce a genetically identical copy of an individual for tissue or organ transplantation. And disadvantages of animal cloning. First, we reduce variety in a species. Cloned animals have short lifespans. Cloning is regarded as immoral by many people. Many, even thousands of eggs may be reared to generate one viable clone, a wasteful, expensive process. And the task for you, put the stages of the two methods of animal cloning in order. Extra challenge. Why do you think animal cloning is more difficult than cloning in plants? And I give you a few minutes with picture illustrated embryo transplant. Cut and stick the stages in the correct order. And next, adult cell cloning. Cut and stick the stages in the correct order. And in the groups, you discuss and fill in the table what are the benefits, problems with each method. Plant cloning, benefits and problems, and animal cloning, benefits and problems. And now we discuss ethics of these methods of reproduction, plant and animal. Is cloning right or wrong? Highlight which statements are for cloning and which are against. Write a short paragraph of your opinion, giving reasons for your opinion. With your opinion line, and if you agree, you stay on the green region of this line. If you disagree, you stay on the red region of this line. If you don't know, you stay on the center of this line. And then tell the person next to you where you sit on the line and why. And summarize the different types of cloning on your worksheet. You only have few minutes. After this time, your work will be given to partner and they will add to your answer in a different color pen. Summarize today's lesson in one sentence. And now your homework. Describe methods of microclonal propagation of plants. The diagram shows a method of producing a large number of plants which all look the same. Cells taken from the bud can be split into many groups. Each group of cells is then grown under the same conditions. And your tasks. First, what do the scientists call organisms which are produced from one parent and which all look the same? Draw a ring around one answer. Give two reasons why plants produced by these methods will all look the same. And next, busy lizzy plants produce flowers with many different colors. A gardener wants to produce busy lizzy plants to fill a flower bed in her garden. She decides to grow them from cuttings rather than seeds. give one condition that she should supply to the new cuttings so that they grow well. Busily the plants can produce flowers which are white, pink or red. A gardener wants to grow a display containing all three colors of flowers. Give one advantage and one disadvantage to the gardener of growing busily the plants from cuttings rather than seeds. Advantage and disadvantage. And today's lesson you will find on the Moodle НИШ Education. And thanks for your attention.